And now I will speak about microbial communities and how we can study microbial communities and especially their adaptation capacities to assess the effect of chemical pressure in different environments. So normally I'm working with aquatic ecosystem, but I will try to also discuss about terrestrial ones. So just a rapid presentation of our team. So we are the aquatic microbial ecotoxicology team in the research unit Riverly at INRAE. So you can see here a picture of our team in January 2024. So you can see that we had a substitution between between Louis and Johanna last year. And we also have a new postdoc, so Vincent, and a new master internship, Darnica, in the team this year. And we are combining microcosm experiments and in-situ surveys to develop tools and study microbial communities, to assess microbial effects on aquatic ecosystems, and also to develop strategies and approaches to assess the ecological quality of this ecosystem in a context of chemical contamination. And for that, we developed close collaboration with colleagues from the Laboratory of Aquatic Environmental Chemistry, including Cécile and Christelle, who are involved in Farmer. So you can see here some non-exhaustive illustration of our research with a lot of field surveys, but also different experimental approaches in the lab. And those are from the last experiment of Johanna. Okay, I don't have to move.
Yes, thank you.
Okay, it's better for the microphone. So some of you work with microbial communities, but not all of you. So I think it can be important to explain why it is needed to study microbial communities. So from the ecological point of view, we have to keep in mind that they are present everywhere, meaning in all components of ecosystems, but also in biological components of ecosystems. They are extremely abundant and diverse. So short quiz for you before starting. So in your point of view, if you have 1 gram of soil or sediment, so you can see here, 1 gram is not a large amount of soil of sediment. How many bacteria can we find? So if you think it's 1,000 bacteria, just put your finger like this one. 2 millions of bacteria or billions. What do you think? I want to see all your finger. Okay, most of you are true, but not all of you, because in 1 gram we will find billions of bacteria representing few thousands or tens of thousands of species, meaning that even if you work with small amounts of soil or sediment, you will be able to assess a large diversity and a large abundance of organisms. So this is really important. And of course, all these microorganisms contribute to a lot of ecological functions that are important for ecosystems. So this is an illustration with soil, with different functions, but we can find the same kind of illustration with aquatic ecosystems. So microorganisms are needed for the good functioning of ecosystem, but also the good functioning of animals and humans. So regarding the ecotoxicological point of view, because this is the focus of the present presentation, so something that can be viewed as interesting for us is that they are very responsive to environmental changes, including changes due to contamination. Also, since they are diverse, they exhibit a wide range of sensitivity to toxic stresses. So of course, it depends on the type of substance, the type of microorganisms, the kind of species, and so on. And also, they are able to adapt or acclimate to contamination at different scales. So from the individual scale to the community scale. So Today I will mainly talk about adaptation at community scale. So why are they able to adapt quickly? Mainly because they have very short generation times, from a few hours for most of the bacteria to a few days for microorganisms such as algae or some fungi. They also exhibit a high plasticity, so physiological or genetic plasticity, so they can also adapt their way of life or their genetic material. And also something that is really important where we are talking about adaptation at community level is that there is high functional redundancy in microbial communities, meaning that generally ecological functions are performed by different kinds of microorganisms, meaning that even if you have change in diversity, it doesn't mean that the community will not be able to function normally. And in our environment, generally, we can observe a chronic and low dose exposure to different kind of toxicants, and this kind of exposure it can promote adaptation for microbial communities. Because if you have acute exposure, of course, you can impact strongly the community, and it's not adaptation; it's a strong impact and you can lose a lot of species and a lot of functions. But with chronic and low dose exposure, you can observe gradual adaptation within the community. So the question is, to respond to the title of the presentation, how can we detect and quantify this kind of adaptation? So one strategy is to use the pollution-induced community approach. So I will use PICT in my presentation, but PICT is for pollution-induced community approach. And just a reminder, this is not a new tool, because the first publication about PICT was published almost 40 years ago now. So you can see, so it was developed in Sweden by colleagues that are now retired. But Hans Blank collaborated with us several years ago. We tried to develop a cost project. This is a European project because the objective was to standardize method for PICT, but unfortunately we were not able to obtain funding from the European Commission. But Hans Blank and colleagues were very active to promote the PICT approach, but finally there's only a limited number of scientists using PICT. That's why PICT is still a promising tool, even if it was developed almost 40 years ago. But you can see that the scientific community, even if it is a small community, try to be active with the PICT. So you can see here a collective paper that was published in 2016. So you will see during my presentation, when I will mention some papers, I will put the picture of people who are involved in Pharmera. So you will see that the community is working together, but not only for Pharmera. So you can see a paper that was published several years ago, and also a paper published 2 years ago by the Ecotoxicomic Network. And all of this paper mentioned that PICT should be used more and more to improve its capacity to diagnose contamination and its effects. So in the paper of Ahmed Tili and collaborators, there's a synthetic description of the PICT concept. So PICT is based on 2 phases. The first one is the selection phase. So this is during the chronic exposure of the communities, so in the field or in microcosm. So when the community is exposed to contaminants, the community can adapt, and you can observe change in the diversity and also change in community tolerance to the contaminant. So to explain it in more detail, for example, you have this kind of ecosystem with an increase in contamination from upstream to downstream. So you can see the cycle. It can be in the water, but it can also be in soil. So you can decide if you want to work with aquatic or terrestrial communities. So upstream, if you don't have chemical contamination, generally you have high microbial diversity. And if the contamination increases, you can observe change in the microbial diversity with a decrease in the sensitive species and generally an increase in the tolerant ones. So normally, if you have a decrease of sensitive species and an increase of tolerant ones, it means that your community as a whole will increase its capacity to tolerate the toxicity of the contaminants that are responsible of the change in the diversity. So the challenge is to be able to measure this tolerance. So to that, we are performing acute toxicity tests. So exactly the same kind of tests that you use during this week, but these tests are developed with the entire community, meaning that we will collect the community and we will perform the test directly with the community. So this is an example with periphytic communities, but you can do that with soil or sediment communities. So you expose the community for a short time to increasing concentration of toxicant, and then you can obtain a dose response curve as this one and as the dose response curves you have seen this week. And you can compare the tolerance, and generally this is done using the effective concentration, the median effective concentration, so the ECE50. So the one challenge is to select the functional parameters that can be used to perform this test. So could you provide me some example of parameters that can be used, functional parameters that can be used for this kind of toxicity test? Any ideas? Also, of course, some of you have already performed toxicity tests.
Maybe carbon and nitrogen respiration.
Yeah, no? Yeah, it's possible. Yeah, Louis. Photosynthesis. David. Yeah, it's the same. Other ideas? Enzymatic activities. Okay, so you can use different kind of function, but you have to define the couple. So the combination, what is the function that is responsive to the toxicity of the contaminants? Because generally we don't know. If you work with pharmaceuticals, pharmaceuticals are not known to be toxic for photosynthesis or for nitrification, for example. So you have to firstly to test and to select the most sensitive parameters. That's why actually there are only limited possibilities to perform picked approach, because it takes time to test the different toxicants and different functions to find the good combination. And I think that something that is missing in the papers that we have written several years ago, and I think this is important, the last phase is the interpretation phase, because today it is still difficult to compare the results between different rivers or different soil and so on. There's a lack of information about the influence of confounding factors. For example, what about co-tolerance in a context of multiple contamination or something like that? And also there is a lack of reference framework, meaning that today we need a reference station, a contaminated one, and we have to compare between the two. But it's not possible, or it's very difficult to compare results obtained in a river with results from another river. So for the water agencies, for example, they say this is not an operational tool, because it is not always possible to have a reference station in the same river or in the same geographical scale and so on. So I think this last step is also important. So just to show you how it can be used, I will show you some example of how it works. So first example from the Ardier-Morcy area in the French Beaujolais area. So this is a famous vineyard area in the center of France. So it was previously mentioned by Christelle, Cécile, and also by Guillaume. So this is important to work with this kind of study site, because we have some long-term socio-ecological research study site in France, and meaning that you can perform research for a long time, so meaning several years or sometimes a longer time, and you can work with a lot of scientific teams, and you can improve your knowledge of the site, and you can also assess the temporal changes and so on. So this site is an example of territories belonging to this long-term socio-ecological research area. So in this area, you have a small river here. Okay, well, okay, you can see. So which is called the Morcy River. So here you are upstream, and here you are downstream. You can see in the pictures that there's a lot of vineyards, and in vineyards we are using pesticides and metals, especially copper. So from where is yeah, okay, from upstream to downstream, we can observe an increase in copper concentration and an increase in pesticide concentration, including the herbicide Diuron, which was used in France until 2008. And 20 years ago, several studies were performed to show that downstream microbial communities, so downstream microbial communities that were collected. Oh, I cannot find the. Okay, that were collected here in the station number 3, were more tolerant to copper and diurons than communities collected upstream. And we also showed that microbial diversity also differed between the different stations. So to illustrate that, this is one of my first papers when I was a postdoc at INRA. It was Semagref during this year, because Semagref as becoming ISTEA and then INRA. And so I collected periphytic communities, so periphytic biofilms, monthly in the 3 stations. So here you can see only the result obtained at the intermediate and downstream station. And you can see here the tolerance measurement, so the EC50 value. So if you have higher EC15 value, it means that your community is more tolerant. So here, I tested the toxicity of the herbicide Diuron by measuring photosynthesis on the communities. And you can see that samples collected in downstream were always more tolerant than upstream communities. And you can see also temporal variability, which is due to the temporal period of the use of Diuron, because Diuron was mainly used in spring. So you can see that communities samples after the use of Diuron were more tolerant than communities samples at the beginning of the year when Diuron has not been used. So, as previously mentioned, Diuron was banned in France in December 2008, and we observe a progressive decrease in Diuron concentration in the river. So you can see that this is not an immediate decrease. It took time to decrease, but you can see that during the first year, it was possible to observe concentration higher than 10 micrograms per liters, and 3 years ago, it was generally lower than 0.1 microgram per liter. So clearly we observed a decrease. So I continued to collect periphytic communities monthly during almost 4 years. So it was quite a huge work, and to measure the tolerance of the communities. So you can see that upstream, so the box plots were done using all the values that were obtained during the year. So these are box plots prepared using the 12 values from each year. So you can see that in green, the tolerance value were quite stable between the year, and you can observe a decrease in the tolerance to diuron at the intermediate and downstream station. So clearly, we observe the progressive decrease in the capacity of the communities to tolerate diuron, but you have to keep in mind that in the river, only the concentration of diuron decreased because we still observe copper or other pesticide in the river. So you can see that this microbial response is quite specific of the kind of contaminant. Another nice example, so this study was performed by Florian during a PhD in Thonon-les-Bains at INRAE also. She compared results obtained between the result she obtained, the result she didn't participate to the campaign, so to the first campaign, but she used the same method that from first campaign that was done before the 20s, and another campaign that was performed in 2010. And you can see that there was a clear decrease in the concentration of the herbicide atrazine in the Geneva Lake, and also a clear decrease in the concentration of copper. And this was associated with a decrease in the tolerance of phytoplanktonic communities. So the tolerance was also measured based on photosynthesis. So you can see that there is clearly a relationship between the exposure level of phytoplankton to atrazine and copper and their capacity to tolerate the acute toxicity of this contaminant in the lake. So this is a very nice example also of the relevance of PICT approach. And regarding the Pharmera project, what about pharmaceuticals? Because most of the examples are based on pesticides and metals. But finally, there is still a lack of studies with pharmaceutical, even if you can find some of them with, for example, in soil, the antibiotic sulfachloropyridazine, and also work performed by Natalia, Elena, and collaborators with some non-steroidal anti-inflammatory drugs with peak measurement performed using photosynthetic activities and beta-glucosidase activities. So photosynthesis for phototrophic microorganisms, so mainly microalgae and cyanobacteria, and beta-glucose activity for heterotrophic microorganisms, so mainly bacteria and some fungi. And a work that we performed recently in another river, so a river located near from the Lake Bourget, so it's not so far from Lyon. This is a high lake, so I think this is highest lake, natural lakes in France. Lake Bourget with a city of Aix-les-Bains. This is a French Alpes Riviera, so a famous city in France. And we performed a survey, so we deployed some process in the river, and we also collected surface sediment. So you can see here the result of the chemical analysis, which were performed in the lab of Cécile. So you can see a list of pharmaceuticals. This is not important, just to show you that there's an increase in the contamination between the upstream station and the downstream one, which is located just downstream from the city of Aix-les-Bains. And you can see that the contamination in the sediment is not at the same station, because several meters after the downstream station, you have a small port. You can see here a picture, which can increase sedimentation of the suspended matter. So you have an accumulation of the contamination in this section of the river. So we perform PICT approaches in collaboration with Chloe, and we observe an increase of the tolerance of the microbial community. So the example are based on the beta-glucosidase activity. So you can see an increase in the tolerance in the most contaminated station. But of course, I have just selected the most beautiful results for this presentation, because All the results were not as expected, but this is always the case. But in some cases, PICT was really relevant. I will try to convince you about that with another study that was performed by Hélène Rogue, who is a PhD student under my supervision and the supervision of Cécile, with an article under revision with this study. So she performed a study in the same site. So she uses glass slides. So you can see a picture here of the glass slides, who were put in the port and also in the upstream site. So the slides were in the river for 28 days. And then some of the slides were moved from the upstream to the downstream station and from the downstream to the upstream station to assess the change in the tolerance to several pharmaceuticals. And you can see here the results. So you can see the results with two pharmaceuticals, so diclofenac and atenolol, with a PICT measurement based on photosynthesis. So you can see for the two pharmaceuticals that the tolerance level downstream, so in red, were always higher than the tolerance level upstream. So this is clearly due to the increase in contamination from up to downstream. And interestingly, when she moved community from downstream to upstream, she observed a decrease in the tolerance. And when she moved communities from upstream to downstream, she observed an increase in the tolerance. So you can clearly see that tolerance level is associated with the exposure level in the field. And in parallel, we assess the diatom diversity because, as mentioned, the tests were based on photosynthesis. And we know that in this kind of biofilms, most of the photosynthetic microorganisms are diatoms. So we assess, Hélène assess diatom diversity using metabarcoding on the RBCL gene, which is a gene specific of diatoms. So you can clearly see some differences between upstream diversity and downstream diversity. You can see change in the diversity with the X downstream and X upstream communities. So this can be illustrated by NNDS analysis. So you can clearly see when you compare here the upstream communities and the downstream one that they are clearly different. And you can see a temporal variation in the X downstream, X upstream. So the arrows are to show the temporal variation. So you can see that the upstream community is quite stable during all the survey. So all the survey, it means 70 days, okay? So you can see the upstream community is quite stable. Downstream, you can observe a marked temporal evolution, and you can see that at the end of the survey, X downstream were close to upstream communities, and X upstream were close to the downstream communities. So you can see an evolution of the communities according to the contamination. So if we analyze the strengths and limitations of the PICT approach in practice, we can say that PICT is relatively simple and low cost, and the deployment and sampling is easy. There is also low analytical cost because it's not too costly to perform the toxicity test. It can allow to focus on specific substances or substances classes. So this is quite specific, a specific response. It can allow us to link exposure to effects on natural communities. So it is not always easy to clearly establish link between effects and community exposure and effects. And also, PICT is still under ongoing development to develop new tests and to cover more substances. But an important limitation is that there are a lot of technical constraints with PICT because you have to work with fresh samples, because you have to be able to measure activities, meaning that you have to have fresh samples, and the tests are generally time consuming. So we need to develop automate tests and probably high throughput methods There's also a lack of a standardized method because all the labs use their own methods, so it's sometimes difficult to compare the different results. And there's a lack of analytical reference framework, still a limited number of substances that are covered, and as previously mentioned, we also have to improve our knowledge about the influence of different kind of environmental factors. So this is okay for the picked approach. I will now move to another capacity of adaptation, which is based on the biodegradation capacity of microbial communities. So, of course, there are a lot of studies dealing with biodegradation, but today the question is, how can we use this function to develop functional indicators of contamination and effects? So I'm working in this topic since my postdoc, with a close collaboration with colleagues from Dijon, including Marion and Fabrice, who are in Pharmera. And the first study we performed was also in the Mersey River. And during these studies, we collected soil and aquatic samples. So in a vineyard, in 2 grass buffer strips located downstream from the vineyard. One buffer strip receiving all the water from the runoff, and another one which was not directly exposed to the vineyard runoff. And we also collected sediment and periphytic communities upstream and downstream from this vineyard parcel. We measured the capacity of the microbial communities to degrade the herbicide diuron. To do that, we used 14C label diuron. So you can see here the chemical structure of the diuron, and you can see here in red the label carbon. And the sediment, the soil, or the biofilm were incubated with the label diuron, and then we measure the production of 14C labeled CO2, which is directly produced from the mineralization of the diuron. Meaning that if you are able to detect and quantify 14C CO2, it means that the diuron has been mineralized. So here are the results. So regarding the soil communities, we observe the highest diuron mineralization capacity in the vineyard soil, and also in the grass buffer strips that was exposed to the runoff. And in the aquatic communities, we observe degradation capacities only in the downstream communities. So it shows that clearly the biodegradation capacities was related to the exposure level in the field. And we demonstrated that previous diuron exposure in the field led to the stimulation of the biodegradation capacity. So again, I studied the effect of the ban of diuron on these capacities. So so the question was, is it possible to observe a decrease in diuron biodegradation potential following the decrease in diuron concentration in the river? So to do that, I only work with sediment communities. So you can see here the mineralization kinetics of diuron in the sediment during 4 successive years at different periods, so spring, summer, and autumn. So you can see that upstream, finally, we observe low mineralization potential and high temporal stability over season and over years, meaning that upstream sediments could be considered as robust reference samples. Yeah, the result obtained in summer, but it was exactly the same for spring and autumn, but it was easier to present, to only present one season. So you can see, as previously mentioned, that the results were the same for all the years upstream, and you can clearly observe a decrease in the mineralization capacities at the intermediate and downstream stations between the different years. So clearly, based on the previous question, the ban of diuron led to the decrease in diuron concentration in the river, which led to the decrease in diuron biodegradation potential in the sediments. And again, what about pharmaceuticals? It is now well known that environmental microbial communities can adapt to the biodegradation of pharmaceuticals, including some antibiotics. So it was not expected because antibiotics are normally used to impact microbial communities, but now we know that microbial communities can also degrade some antibiotics. So one of the first work about that was performed by Edtop, who was in Lyon during the kickoff meeting. And he studies sulfametazine degradation capacity in soils that were treated during several years. So you can see 12 annual application of sulfametazine at different concentration. And you can see in the figure that the biodegradation capacity clearly increased in the soils that were treated with the highest concentration of sulfametazine. And so now we know that antibiotics degradation is possible. So there is clearly a growing number of studies on this topic. So this is also our case. So we have a study that was also performed by Hélène during a PhD with the help of Louis during his master internship. So she developed a microcosm experiment using sediment from the TA river that was previously mentioned. So using microcosm, so you have the picture. She tested 5 concentration of sulfametazine. So the antibiotic sulfametazine. So you can see here the concentration of sulfametazine in sediments, which were measured at the start and the end of the experiment. So at D0, day 0, and day 47. So we observed a strong decrease in sulfametazine concentration at the highest concentration. And in parallel, thanks to the collaboration with Marion in Dijon, we were able to show that this was partially due to the increase of the capacity of sediment communities to degrade these antibiotics. You can see with the red and the orange curves that at the 2 highest concentrations, the communities developed capacity to degrade sulfametazine. And this change in the biodegradation capacity was also associated to change in microbial diversity, especially bacterial diversity. You can see on the right of the figure here, no, not here, so in the figure directly, that the community diversity clearly changed according to the exposure level. So the bioinformatics analyses were performed by Louis. So again, strengths and limitation of this kind of approach. Generally, it's not too costly to assess biodegradation capacity. Again, the response is very specific of a kind of substance. It can help link exposure to effects, and methods are still under development. But again, there are some significant technical constraints. The first one is the use of radiolabel substances, because this is expensive and subject to numerous constraints, because it's not always possible to use this kind of substances. And also, as you can see here, for example, the kinetics for biodegradation are generally quite long. So you have to wait for several weeks before obtaining the result of your study. So a possible alternative is to use molecular approaches to study functional genes that are involved in the biodegradation. So this is the last part of my talk. I will give you some example of the use of functional genes. So I mentioned degradation genes, and in your point of view, which kind of genes could be also studied to assess the adaptation of microbial communities to contaminant? Now, clearly, adaptation to contamination. A specific gene, kind of genes. Yeah, resistance. Not specifically antibiotic, but every kind of contamination resistance. Okay, so some examples. So I will I will come back again to the experiment of Helen. So in parallel of measuring the biodegradation capacity, we also quantified one gene that is known to be involved in the sulfametazine degradation, and you can see that we were able to show that the quantity of this kind of gene increased with the increase of the capacity of degradation. So you can see that it's probably easier to quantify the genes than to perform the biodegradation kinetics. But one limitation is that this kind of genes are generally not abundant in the community, so it's not always possible to detect them in the community according to the quantification level and detection level. Another example with the study which was performed several years ago with the degradation of diuron. We know several genes involved in this degradation, so the genes are named PUH-A and PUH-B. And for example, we expose the vineyard salt to different concentration of diuron, and you can see here that the soils that were exposed to the higher level of diuron exhibited the higher capacity to degrade, and they also exhibited the highest abundance of this kind of gene. So sometimes it is possible to use degradation genes, and also regarding resistant genes. So we mainly work with EdTop for that. So we assess the abundance of resistant genes in the TA river. So you can see the green circle is a quantity of resistant genes in the upstream station, meaning that if you are, for example, here, you have 1,000 more resistant genes than upstream. Okay, so you can see, so for almost all of the genes, the quantity was always higher and strongly higher than the resistant genes quantity upstream, both in periphytic communities and in sediment communities in the TA river. And it was the same in the Ardier river, both in periphytic and sediment communities. So you can see that clearly in contaminated river, you can observe an increase in resistant genes. The question is the increase due to the contamination, to the exposure of the communities to the pharmaceutical in the river, or is it a contamination from the terrestrial environment? Probably both of the two mechanisms. You have a direct input of resistant genes from the terrestrial environment, and probably the contamination in the river can help can help the population harboring this kind of genes to develop in the natural communities. And another example, the last one, and it will be the end of my presentation, a survey that was performed in Geneva Lake several years ago when I was hosted by Benoit in his lab at the Ecotox Center. So we performed a sampling survey in different stations, and we observed a clear increase in the quantity of several resistant genes in the most contaminated area of the lake, so near from the city of Lausanne. So this is clearly the hotspot of contamination in the lake sediment, in the Geneva Lake, and also near from the Rhone River. So we don't know if it's due to an input of resistant genes from the Rhone, or if it's due to contamination by pharmaceutical in this station, because we didn't measure pharmaceutical during this survey. That's why we performed another survey 2 months ago. So a new monitoring campaign with 8 days of sampling, 30 sampling stations, with a lot of analysis. But regarding microbial communities, we will perform picked approach using several contaminants, including sulfametazine. And also we will screen for a lot of resistant genes, and we will also perform metagenomic approaches. So as you can see in this picture, it was a very difficult survey, and we had a very hard condition to work. Yeah. So to conclude, clearly there are some tools and scientific evidence that can help demonstrating that studying microbial adaptation capacity can help to assess chemical pressure and impact. But there are still needs to develop, optimize, and standardize methods, propose strategy to generalize interpretation, better understand the influence of several environmental factors, and also, and I think that this is important to convince everyone that it is important to study microbial communities, to assess the consequences of adaptation at ecosystem levels. Because when you have adaptation, generally you have a loss or change in diversity, and so probably you have effect on ecosystem functioning. But generally we are not able to characterize this effect. So I think we have to work on that. And the last point, it should be important to assess the influence of environmental continuums to be able to differentiate between effect of chemical pressure and influence of inputs from soil or from terrestrial ecosystem or from upstream station and so on. So this is one of the objectives of the PhD project of Joanna. And this is all for me. And thank you for your attention.